Okay, good day, class. Now we are on the last leg of our Innate Immunity series lecture. And if you can observe on the series 1, we discuss more on the humoral part, which is your acute phase reactants, correct? And we all know that acute phase reactants is part of the humoral together with the complement. However, on the complement part, we will have a separate discussion with that one kasi it's a long discussion session. Okay, so in the innate immunity series, lecture 2, we will discuss on the cellular response. Okay, so you have your immune cells. Okay, so the second line of your defense. So of course, with your immune cells, okay, one of their major function is phagocytosis, but not all of them naman are capable. We will discuss that one on the second video of your Innate Immunity series lecture number 2. I would also like to clear things up with this one because the immune cells video lecture is somewhat an introduction. Why? Because I believe that your immune cells are introduced already with your human anatomy. I also teach human anatomy class during my younger days. So I teach human anatomy. So that's the reason why I know that this topic is included already. And then I also teach before immunology and serology with other universities. That's the reason why I know that your immune cells is part of your uh, immunology serology laboratory. Kasi the first topic for laboratory is for the identification of your cells. You need to have this one. And also with your hematology. So hopefully, yeah, it would be easier for you to understand the basic principle and or foundation of your immune cells. Okay, so let's start. Again, so I'm your professor. I'm Alfredo Hine Jr. and you can call me Sir Red. And if you have some questions and or clarification with this video lecture, please don't hesitate to contact your professors so we can clarify the gray areas of your immunology and serology course. Okay, so now let's proceed to your immune cells. So your immune cells are actually have five parts. Okay, so they are classified into neutrophils, okay, eosinophils, okay, basophils, monocytes, and lymphocytes. So the structure are different, so therefore you need to familiarize that one in your laboratory. And then if you will practice your profession in the near future as medical technologists, medical laboratory scientists, this is very crucial class kasi if for example you misidentified your neutrophil as a eosinophil, and then what will be the interpretation? Allergic reaction and or something hypersensitivity, which is not good class. Okay, in or with your basophil to your monocytes, but that's too uh, malayo na yung structure nila class. But there are some students that still miss identify the immune cells. Maybe because in my personal observation, especially with especially now na online, is that the pictures for example, in the Google or in the internet is somewhat different pag nasa actual na peripheral blood. So, I hope that we can cope with that one, and then because this is very crucial. Again, the purpose of this slide is for you to at least understand that we have a major five parts of your immune cells. Then you have your neutrophil, eosinophil, basophils, monocytes, and lymphocytes, and we'll try to cover that one by one. So let's start with your polymorphonuclear cells or PMN leukocytes, also known as your neutrophil. Okay, so it represents approximately 50 to 70%, okay, of the total peripheral white blood cells. So sila yung pinakamadami class. As I mentioned in the previous session, that the population of your neutrophil is divided into two. 50% is located on the system, and then 50% is located to your endothelial linings. The reason with that one is that I also explained with the process of your inflammation so that the migration will occur faster than normal. So in structure class, these are round 10 to 15 micrometer in diameter with a nucleus that has between 2 and 5 lobes. Okay, this is the reason why they are called poly. Okay, polymorphonuclear cells. Poly ibig sabihin madami class. Okay, polymorphonuclear cells. Okay, so this is your neutrophil. So they're somewhat large, around 10 to 15 micrometer in diameter. So please take note with that one. So if you can observe in your red blood cells on the peripheral blood, in the picture, if you can observe here, the red blood cells are somewhat smaller compared with your PMNs or polymorphonuclear cells. I want you to show the lobe. The lobe is this one. Allow me to have my cursor. Plus, so you have this one. This is your neutrophil, right? So may lobe siya 1 and then 2, and then 3, and then small 4. So hanggang abot siya class. Kasi hanggang ano daw, ilan yung lobes? 2 to 5 daw siya. Then you have your 2, 1 lobe, and then 2 lobes. Then they are granulated. And then the granules will explain that one. So the neutrophil or your PMNs, they contain a large number of your neutral staining granules, which are classified as primary, secondary, and tertiary granules. Take note of this one class. Okay. For your primary granules, they're also called your azurophilic granules class. Okay. One is your myeloperoxidase, you have your elastase, you have your lysozymes, you have your protease 3, you have your cathepsin G, and you have your defensins. Okay, so the primary granules class, their main function is for antibacterial. This is the reason why your neutrophil is the primary immune cells to fight against your bacteria, because their primary granules main function is to kill your organism. And then for the secondary granules class, you have your collagenase, you have your lactoferrin, you have your lysozymes, then you have your NADPH or reduced nicotinamide adenine dinucleotide phosphate oxidases. Okay, so with the secondary granules, these are also, like for example, lysozymes is a good antimicrobial or antibacterial activity. So your secondary granules can also used for your antimicrobial activities. However, other part like for example your collagenase, your lactoferrin, okay, so these are also a component of your WBCs. When over express, it can damage the human host and your human tissues. Okay, take note of that one. And then you have your newly discovered tertiary granules, you have your gelatinase, you have your plasminogen activator, and you have your acid hydrolysis. Okay, so part of this one is still active against your microorganisms and or can damage your tissue. Okay, so if you can recall with the discussion with the previous session that I always mentioned that your immune cells usually overreacts. So therefore, your APRs will try to neutralize that one. And normally, half of the total neutrophil population is found, ayan, lumabas na class in my PowerPoint. Okay. So normally, half of the total population is found in a marginating pool on blood vessel walls, while the rest are circulate freely for approximately 6 to 10 hours. Okay? So again, I mentioned this one already repeatedly that the main function why the 50% of the 50% of your population of your neutrophil resides to the blood vessel walls because it can facilitate faster your inflammation. Okay? Okay. Next, you have your eosinophil. Okay, your eosinophil is somewhat almost the same with your neutrophil, around 12 to 15 micrometer in diameter. And they normally make up between 1 to 3% matching class, from 70% na neutrophil to 3% na class, eosinophil in non-allergic person. So if you are non-allergic person, then for example as normal, then your neutrophils should be around 1 to 3. Okay? So their number increases in an allergic reaction or in response to many parasitic infection. Okay? So, pag may mga parasitic infection, usually your eosinophil will increase, your neutrophil will slightly increase also. Yan. Okay, the nucleus is usually bilobe or ellipsoidal, and is often eccentrically located. So, eccentrically located. So, nasa ano daw sa class? Yan. We can observe here, yan, with the bilobe siya. And then you have your granules also. Large granules. Yan. Tapos, compare kanina with the neutrophil. I want to show you the neutrophil. Azurophilic granules sa class, so color blue. Yan. Color blue yung granules niya. Yan. Okay? So, compare here with your eosinophil. I want to show the eosinophil kasi eosinophil is very nice. That's one. And then you have your red blood cells. Okay? And then you have your bilobe. Okay, bilobe na location, ano naman class? Eccentrically located. So the primary granules contains your acid phosphatase. You have your aryl sulfatase. Okay, aryl sulfatase, very good enzyme for your Mycobacterium tuberculosis. If your eosinophil also will increase if you have your Mycobacterium tuberculosis. Then you have your eosinophilic specific granules or major basic protein. You have your eosinophil cation proteins. You have your eosinophil peroxidase, eosinophil derived neurotoxin. So these uh, uh, components are generally termed as your eosinophil specific granules. So these cells are capable of phagocytosis together with your neutrophil, take note of that one, but are much less efficient than your neutrophil. Okay? So, ibig sabihin class, if they have a parasite, di ba parasite are two pigs, for example. Okay, tayo ng ano. This one is your parasite class. Okay, this one is your parasite. And then you have your cell. This one is your cell. This one is your eosinophil. Then your eosinophil will try to eat that up. Pero hindi niya makain class. Hanap ako ng ano. Like for example, this one is your parasite. This one is your parasite. Diba? So this one is your parasite. And then this one is your neutrophil. And then your neutrophil, as a phagocytic cell, they will try to eat up. Tama? However, the eosinophil will try to check, masyadong malaki. Hindi ko kaya ito kainin. So what should I do? Now, your eosinophil will release enzymes, yung na-mention ko kanina na mga enzymes, and the enzyme will try to kill the parasite in half. So, ano niya class, one by piece and piece niya class into small piece, and small piece. So if you have this one, very small na class, and then the neutrophil, the eosinophil now can actually eat up. Kasi maliit na sila. O diba? Ganun katalino yung mga eosinophil class. Okay? Then their most important role in your neutralizing, their most important role is neutralizing the basophil and mast cells product and killing certain parasites, as I explained earlier with the killing certain parasites. Ayun, ito yung sinabi ko kanina class with your mechanism of action of your parasite killing. So since the helminths or the parasite is too big, so what they need to do is to release your molecules. And then also, it is antibody mediated. Your eosinophil requires antibody. So the antibody will become opsonine, and then the neutrophil, specifically here, kasi IgE siya class. Here, this one is IgE. If you can observe, then you have your IgE receptor. Okay? So your eosinophil have the IgE receptor that will bind to the Fc portion of your IgE. Then they can now engulf. Hindi pala nila ma-engulf class kasi masyadong malaki. Then they can release in class. And then, pag maliit na yung mga parts ng parasite, then they can now engulf and phagocytize. Diba? They're very clever class. Okay? Always take note that your eosinophil contains EFC epsilon receptor class. If you can observe in the PowerPoint, they have an FC epsilon receptor. So if you have your antibody, this is your FAB. Okay, this is your FAB. This is the area where the with the antigen will bind class here. Then you have here, this area class is your FC region. Your FC region, yun yung puwet ng antibody class. This is the area where the immune cell will attach. Why? Because the immune cells have the FC epsilon receptor. Okay? Then let's move to your basophils. Okay, less than 1% of all circulating white blood cells. Okay, less than your eosinophil. Okay, the smallest of all the granulocytes, around 10 to 15 in diameter, and contains coarse, densely staining, deep bluish purple granules. Okay, so if you can observe on the peripheral blood slide, that are often obscured the nucleus. So we cannot actually observe the nucleus with your basophil. So the constituents of these granules are histamine and then small amount of heparin and eosinophil chemotactic factor A because if kung activated yung basophils class, they will release a granules. You have your eosinophil chemotactic factor A. Pag sinabi natin chemotactic, ibig sabihin yung class, tigtawag. Okay, so it means that if the basophils are there and in very small amount, so they need syempre best friend. So with your basophil, they will try to release your chemotactic factor A, then it will try to call out your eosinophil, then your eosinophil will help your basophil, di ba? Because they're very friends sila. And also with the histamine, we'll explain the histamine. We will discuss your hypersensitivity. But for now, at least you have your histamine. And then you can now correlate the histamine to your antihistamine, diba? You take your antihistamine because there is a overproduction of your histamine. But take note with this one class, your basophils will mature to your mast cells. And your mast cells will release a large amount of your histamine, and we call that one degranulation. So your basophil also have an IgE, okay, interaction, can immunoglobulin form in allergic reaction, okay. So this is somewhat the same with your this is somewhat the same example of your eosinophil, and binds readily to basophil cell membranes, and granules release their constituents when they contact an antigen. So your basophils will mature to your mast cells. Your mast cells will release your mediators, inflammatory mediators. Tawag naman natin dyan class mediators. It's a general term. Okay, one best example of your mediators is your histamine. Other naman class, you have your lipoxygenase A, you have your prostaglandins, and then a lot of new terms with that one, but we will discuss it pagdating natin sa hypersensitivity. Yeah. So that is how your allergic reaction occurs, class. And then you have your mast cells. Okay, these are the tissue mast cells, resemble basophils. They are a connective tissue cell of your mesenchymal origin. Okay, they are widely distributed throughout the body and are larger than your basophils with a small round nucleus, okay, and more granules. Okay. So your mast cells and your basophils are somewhat related. So ano sila class? Mag-ig-agaw. Okay, they are cousins. So they are responsible for your allergic reactions. And mast cells, as we mentioned here, class, they are located on your connective tissue cells. So therefore, it would be very difficult to find that one on your peripheral blood. Okay, smears. Okay, so mostly yung nakikita lang talaga natin is your basophils. So your mast cells have a long lifespan of between 9 and 18 months. Imagine class 18 months. And then the enzyme content of the granules help to distinguish them from basophils because your mast cells also contain your acid phosphatase, you have your alkaline phosphatase, and you have your proteases. Again, you have your ACP-ALP, and you will try to discuss that one more deeply on your clinical chemistry with your enzymology. The mast cells like the basophil, okay, plays a role in your hypersensitivity reaction by binding IgE. Okay, so ang pangit naman, di ba, class, if I will try to ask what type of hypersensitivity, kasi we have four types of hypersensitivity, di ba? And you don't know that one yet. But for the sake of this presentation, with the IgE mediated, so this is usually your type 1 hypersensitivity, and it's fast. Yan. Okay, so I know that the topic of hypersensitivity is far beyond this area pa. However, we can correlate that one. That's the beauty of your immunology class, because each topic has a relationship and connections. So that's the reason why you need to understand per session. Why? Because you cannot understand the more complicated part of our immunology if we cannot grasp the basic foundation of your innate and your adaptive immunity. Okay, so let's move to your monocytes. Okay, monocytes or your mononuclear cells are the largest, okay, largest cells, around 20, around 12 to 22 in your peripheral blood. Okay, one distinguishing features is irregularity fold or horseshoe-shaped nucleus. Ayan, so if you can observe on the picture, on the nucleus, right? So you have your horseshoe nucleus, like, okay? The NC ratio also is somewhat, the NC ratio of your monocyte is 2:1. Mas malaki dapat yung nuclear to your cytoplasm. The NC ratio will be explained to your laboratory. Okay? Because the NC ratio is a tool or parameter to identify your lymphocyte from your monocyte. Okay? Like for example, ano pala yung NC ratio pag tinatanong ka sa defense ng laboratories? What's the NC ratio? How do you how did you decide that this cell that you found in the microscope is a monocyte? And you can like mention, sir, it's because of the NC ratio. The NC ratio that I can see now is 2:1, and this is a typical characteristic of your monocytes. Yan. So that's how you do it in the laboratory. So the abundant cytoplasm stain, a dull grayish blue, and has a ground glass appearance. Take note with this one, class. Usually lumalabas sa board exam. Okay, ground glass appearance due to the presence of a fine dust-like granules. Okay, they're still granulated, class, pero hindi lang natin talaga sila nakikita. Okay, these granules are actually of two types. So that contains your peroxidase, your acid phosphatase, and your aryl sulfatase. Again, your aryl sulfatase is a good enzyme for your Mycobacterium tuberculosis. Then these indicate that these granules are similar to the lysosome of your neutrophil. Tama, diba? Kasi diba, as I mentioned earlier, it's almost the same. Like for example, you have your aryl sulfatase and you have your lysosomes. This is the reason also why, class, that your Mycobacterium tuberculosis, if you have a chronic inflammation, you have a chronic infection, your immune cells, specifically your neutrophil, will still be decreased, but your monocyte is increased. So the other type of your granules may contain your beta glucuronidase, your lysozyme, and lipase, but no alkaline phosphatase or ELP. Okay, digestive vacuoles may also be observed in the cytoplasm that may indicate a active phagocytosis. Then they stay in peripheral blood for up to 70 hours, and then they migrate to the tissue and become known as your macrophage. Ayun. So, they'll try to circulate sa peripheral blood for 70 hours. Tapos pag wala silang makita, boring masyado yung sa blood, I'll go to the tissue. Then they become more stronger. Okay, have your macrophage. Okay, your macrophage is a potent APC or antigen presenting cell. Okay, compared with your monocytes. Maybe because they, yung galamay nila, class, with the macrophage, they can easily, like for example, this is the this is the bacteria class. Ito. Then this is your macrophage. Kasi they have pseudopods, so they can easily like get the bacteria and then engulf. And phagocytosis. We'll discuss the phagocytosis after this video lecture. And again, so if you have your macrophage already on your tissue, all tissue macrophages arise from your monocytes, okay, which can be thought of a macrophage precursor. Okay, as the monocyte matures into a macrophage, there is a increase in endoplasmic reticulum, lysosomes, and mitochondria. That's the reason why they are more active. Then, if you can observe here, more granules, more vacuoles, an indicative of phagocytosis. Unlike your monocytes, okay, your macrophages contain no peroxidase. Okay, that's the main difference, class, of your macrophage and your monocytes. So we have different macrophages, class. Their name depends on the location of the body. So, for example, you have your blood for your monocytes, you have your bone marrow for your monocytes, you have for microbial cells for your CNS, kidney for your mesangial cells, your Kupfner, so di ba masyadong human anatomy class. Okay, your liver for your Kupfner cells, your alveolar macrophage or your DAS phagocytes for your lungs. Then you have your lymph node for your lymph node macrophage, spleen for splenic macrophage, synovial fluid for synovial A cells, and then you have your connective tissues for your histiocytes. Okay? So again, the purpose of this slide is for you to understand that the macrophage is still macrophage. However, their name differs depends on the location of the macrophage. For example, kung ang macrophage nasa liver, then they are called Kapner cells. Okay? Take note with that one. And then, you need to at least memorize this one by heart kasi possible na lalabas sa quiz and sa exam and of course, pagdating sa board exam. So their functions include microbial killing, of course, because they are a potent phagocytic cell, tomocellular activity, intracellular parasite eradication, and of course, your phagocytosis. Secretion of the cell mediators, together with your interleukin-1, interleukin-2, interleukin-6, and your TNF. And antigen-presenting cells. They are APC, antigen-presenting cells. The most potent antigen-presenting cells class is dendritic cell, then followed by your macrophages, and then the lowest one is your B cells. Your B cells is the antigen-presenting cell class. Yan. Pero the function of your antigen-presenting cells of your B cell is not a main role. Kasi their main role is for the stimulation and production of your B cells through the formation of your plasma cells. Okay, formation of your antibodies through the evolution of your B cells to your plasma cells. And tadan, so you have your dendritic cells, kaya as I mentioned earlier, the most potent APC, your dendritic cells are named dendritic cells because it resembles nerve cell dendrites. So their main function is, of course, phagocytosis, and also APC, ibig sabihin class, antigen-presenting cell, they will present it to your helper T lymphocytes, tama, to initiate the acquired immune response, which is the adaptive arm of your immunity. They are the most potent phagocytic cell in the tissue. Yun. Okay, take note with that one. Kasi you have your dendritic cells, number 1. Number 2, macrophage. Number 3, you have your B cells. Okay? And that's it for your immune cells class. And which is the second line of your defense. The first part of our Innate Immunity series, Lecture 2, video lecture. So I hope that we have a good reinforcement of your immune cells. Sorry, class, with the batch 2019. I hope that with that one, you have a good grasp of a good foundation class of immune cells that can help with your laboratory and also to cover the innate immune response. Okay? So if you have some questions and or clarifications, please don't hesitate to contact your professors so we can clear up the gray areas of your immunology and serology. And thank you for listening, class, and then God bless. Okay, so again, you can refer my video lecture with your reference book. Bye, class. Bye bye.